Okay, so our next speaker, and I'm very sorry for Andrew because he's only worked with one mentor in the center. So, and I was joking to him about this before. So Andrew Lowell did his PhD at the University of Pennsylvania with Maria Kozlowski. And then went to work with David Sherman again. So David is quite an influence in today's program at the University of Michigan. And after quite an extended time there, he began his own independent career at Virginia Tech. And we're very much looking forward to hearing about your program. So the title is Pioneering New Routes for Antibiotic Development. Welcome, Andrew.
Thank you, Hugh. I appreciate it. Can everybody see this okay?
Yes.
Yes. So thank you so much for inviting me today to talk. And as Hugh introduced, I started doing total synthesis work with Marissa at UPenn, and then I moved on to a center professor in David Sherman at Michigan. And what we're going to talk about today, if this slide decides it wishes to advance, there it goes, is microbial resistance. And so our group right now is really focused on antibiotic development. So these are some of the statistics that we're seeing right now for antibiotic resistant bacteria and the deaths that they cause. And having just finished one pandemic, we can start to put some of these numbers in perspective in terms of how many more deaths something like this will cause relative to sort of our baseline level of life cycle. And the statistics are predicted to get a whole lot worse in the next 30 years. And so unlike sort of the acute experience we just had, this is something that's spread out, it's among everywhere, and it's really important to keep developing new and better antibiotics in order to prevent unwanted deaths from happening to people. The assay shown here in the background is called a distaffusion assay. And it's really a quick and nice way where you can qualify how much activity a particular antibiotic has. So this material over here on the left is a bacteria that's susceptible to antibiotics. The one on the right is a strain that started to gain resistance. And so that can be really problematic if you're sick with one of these and someone gives you a drug and it's no longer effective. So the golden age of antibiotics kind of started in the 40s through the 70s, and we discovered a whole bunch of new natural products, and we started turning these into new drugs. And you'll notice there's a big gap here starting in about the 70s, which wasn't because we weren't finding new things that killed stuff. It was because we were finding the same things again. So we were still looking for natural products in order to make these materials better, but unfortunately we kept finding the same stuff. So we shifted gears to some combinatorial-based synthesis, and we got some new drugs out of there, but not nearly as many that were fast enough developing in order to beat back some of the antibiotic resistance that was developing to these drugs. So on the bottom of this, we can see when antibiotic resistance started to be observed compared to the deployment of these particular antibiotics that we discovered. And so this gets into what our group does. And very similar to what Allison talked about. So we'll sort of give you the same thing again and really try to get you interested in biocatalytic synthesis, is we're trying to synthesize natural products, in this case, specifically antibiotics. But we don't only wish to undertake traditional synthesis where we try to build these materials from the ground up. We also very much want to look at the biosynthetic pathway. These molecules shown down here at the bottom are all natural products. They're all produced by bacteria, a fungus, a plant. And so we can investigate how that organism produces these from simple precursors. And we can use that biosynthetic influence to think about how we want to plan our synthesis. Additionally, we can then confirm that biosynthetic pathway. We can understand if these genes really are the ones that make them and add to the literature in that way. Additionally, we can take that pathway understanding and incorporate those enzymes into a chemoenzymatic synthesis in order to make not just our target to test, but to make family members and derivatives as well. So we're undertaking the synthesis and the semi-synthesis of a lot of derivatives of these known antibiotics. And I'm also going to talk a little bit today about where all of these projects are leading to, which is the formation of bidentate antibiotics. But before we get there, I do want to talk a little bit about the importance of the CCHF. So when I was a graduate student working on a total synthesis before the center had started, we were trying to synthesize this natural product, purpuromycin. And it took us a lot of trial and error to get sort of to our high water mark, which is this compound shown here, 66. And the tricky part of this synthesis was we needed to form this spiroketal here in the center of purpuromycin. Now, what was at issue was whenever we tried to do it, we would see elimination from these hydrogens in blue to form a benzofuran instead of the product that we want. And so we went back and we thought about how to suppress that. And we ended up putting a ketone where those hydrogens were so it couldn't eliminate. But in order to get that ketone in, we had to remove one of the phenol groups that would be on this naphthalene portion. We were running into gearing effects where we couldn't get everything on with all of that functionality around there. You know, this naphthalene is substituted 7 times. So we removed one of them. And so after a lot of steps and effort, we got to this advanced intermediate 66. And now what did we have to do? We had to reintroduce that oxidative functionality. And we weren't able to do that with the literature present at the time. And so I found what there was, I tried it, and I, for the life of me, could not make it work. So in preparing this seminar, I went back and I looked and I saw what's been produced then. And a couple of years later, after this was published, wouldn't you know, someone came out with a rhodium catalyst that would take an aldehyde or a ketone adjacent to a benzene ring, and it would selectively oxidize one of those centers. So to me, this really shows how critical and timely the development of the center was in order to gain us these selective oxidations that we need when we're doing chemical synthesis. So while working hard on this total synthesis, I got really interested in the biosynthesis. So a bacteria makes this. How does that bacteria make this? And this is really what inspired me to get a postdoc with David Sherman at the University of Michigan after leaving Marissa Kozlowski's group and start to focus on understanding biosynthesis natural products and how I can use my synthetic chemistry skills to access these biosynthetic pathways and then the enzymes that are in them in order to make more and complex natural products. So I'm not going to talk about the synthesis here. Really what I'm going to focus on is some of our biocatalytic methods in order to produce these high-value complex molecular targets. So I'm going to start here from this hexaketide, which is something that we synthesized, and we run it through a whole bunch of biocatalysis in order to get these natural products. So I have a really nice animation that shows this, but it's beyond the scope of this. So perhaps some other time I'll be able to share that with you. But polyketide synthases are really enormous biocatalysts. They weigh between about 150 and 200 kilodaltons. And they do an enormous amount of chemistry all on one catalytic platform in this enzyme. So this particular pair here, JuV4 and JuV5, can take this synthetic hexaketide and it will engage a malonyl SNAC and a methylmalonyl SNAC shown in red and green, and it'll actually attach them to it here. It'll adjust the oxidation states of these materials, and finally it will actually cyclize this material into this macrolactone ring. And it does all of this in basically an aqueous solution with a few cofactors. So this is a bunch of synthetic steps that are highly selective and highly stereoselective in terms of what molecules are incorporated and where they go. The bottom portions here are this reaction as it progresses. It runs in water, but we have to add the starting material in DMSO. So it begins cloudy and it ends up coming clear as the reaction goes over time. And that runs in a fantastic yield, upward of 60% for a whole bunch of different synthetic transformations. So with this macrolactone in hand, we could again turn to a biocatalytic process. So some of my coworkers in the Sherman lab, now Dr. DeMars and Sungryal Park as well, who's also a PhD, designed a whole bunch of biocatalytic systems where they knocked out all of the genes except for the sugar production and attachment portion shown over here on the right. So essentially what we could do is we could grow a bacteria. We could give tilactone to that growing culture. And then after the reaction was finished, we could extract the bacteria and we could get out not only the sugar attached, but the sugar having been synthesized by the bacteria. So this is a marvelous demonstration of how you can use biocatalysis, even in a bacteria itself, in order to get the stereoselective attachment of a sugar to this macrolactone ring. And this now gave us our first natural product, M4365G1. Now, this is where the selective CH functionalization comes in the Sherman lab. So we were studying a whole bunch of P450s. There's a couple shown up here in this particular genetic cluster that makes the rosamycin series. And Matt DeMars and now Dr. Jennifer Schmidt is doing a whole bunch more work with those materials. Matt's gone on to graduate. But what we could do is we could take this G1 first natural product derivative, and now we could use heterologously expressed P450s in order to gain selective oxidation all around this molecule. So the first one from the cluster we were looking at was JuVe-D, and that would selectively install an epoxide at this gamma delta position of this dieno. And so that was pretty amazing in order to get that functionality in. Matt had previously been expressing MIC-C1 and tile-I, and those were selective for the unactivated methyl group shown over here at the C23 position, and the unactivated ethyl group shown up here at the C20 position. So these are really marvelous in their specificity for being able to take this extraordinarily complex molecule and put in a single very selective CH functionalization in order to add complexity to it and allow us to continue our synthesis to make the entire family of this natural products. I would point out that Jen has been pursuing this project further on some work that I'd been doing while I was still a postdoc. And we again have one more imminent publication that's about to get submitted on this same work. So keep your eyes open for that over the next few months. So this is just a summary of everything we were able to make. So sometimes we reverted to chemical oxidation as well in order to generate the aldehyde here. We combined some of these P450s to get double oxidation events on these molecules. And then we were able to test them against a variety of bacteria to see what the activity was. And what we were able to determine was it was really this aldehyde, which was known in the literature, but we confirmed it through this, is really the critical pharmacophore for getting these materials to bind to bacteria and effectively kill them. And so we terminated this project sort of with a activity relationship and testing these materials. So that foray into basically using biocatalysis, specifically CH oxidation, in order to get family members is really what's inspiring my independent work now toward developing new and better antibiotics. And some of these ones that we're targeting here are macrolides that I've worked on before, but the majority of them that we're looking at are ones that I don't have experience with, that we're trying to apply some of these same principles to in order to derivatize. And as I alluded to earlier, what we really want to do is we want to make bidentate antibiotics. And this concept is a subgroup of hybrid antibiotics or hybrid molecules. And all a hybrid molecule is in this point is someone taking two effective medicines and saying, hey, what if we put these materials together? Would these materials somehow work better if we've attached a covalent bond between them? And where I really want to differentiate the work we're doing versus the previous work is sort of the rational approach that we're taking toward doing some of this. So we've seen success in improving properties for these, but one of sort of the lack of progress that's been made is increasing the activity. And in looking back over the literature and trying to assess how people were focusing on these, sometimes they were taking a approach that wasn't always looking at the pharmacophore versus the oxophore. So a pharmacophore is the part that's important for the activity, and oxophore is a part of the molecule, but it doesn't necessarily involve its binding affinity. And oftentimes when we're doing chemistry, we look and we see a unique functional group and we say, hey, it would be really easy to make a bunch of derivatives off that single functional group and I can test all those and maybe the activity will increase. Sometimes you do that, it gets a lot worse, and then you have to use harder to access functional groups as you continue to do your medicinal chemistry. And so most of the previous hybrid approaches have really sort of fallen into this trap. They've linked two molecules together, but they haven't given that linker enough length or flexibility in order to engage multiple target sites. Likewise, they've selected a pharmacophore instead of an oxophore. So despite the fact of having an effective linker there, they've destroyed basically the binding motif of one particular molecule, and it won't bind anymore. Another issue is target separation. So our work is predominantly focused on the bacterial ribosome, which is where proteins are synthesized in a bacteria. Now, there's lots of other wonderful targets in a bacteria, especially stuff like the cell wall, which a bacteria has, but a eukaryotic, a person does not. So you can select for bacteria by picking that target. Unfortunately, if you combine those two compounds together, there's no way you're going to be able to access the interior of a ribosome while you're also trying to target the cell wall, which is at the periphery of a bacteria. So really what we're trying to do is select two antibiotics that we know where they bind. We know what's critical for their binding, and then incorporate a linker to these materials so that they both bind at the same time in a bidentate manner. So as I said, our primary target here is the bacterial ribosome. And it's wonderful. Everything needs proteins to survive. And there's a lot of materials that are selective for the bacterial ribosome over the eukaryotic ribosome. And additionally, there's a whole bunch of co-crystal structures published in the literature that enable us to understand where and how all of these different antibiotics bind. So the ribosome is shown in this slide in the background. It's about 200 angstroms across. And it's got two sort of major regions where antibiotics bind. One is where the amino acyl TRAs come in and deliver the peptides that are going to be included in the protein. The other is where that amide bond is actually formed in the peptide transfer center. And so we have a number of targets within each of those sites. We're not trying to bridge these sites to one another. They're a little bit too far apart, and there's a lot of ribosome between them. So instead, really what we're doing is we're focusing on linking two antibiotics within one or the other of these sites together in order to move toward our bidentate antibiotics. I have a wonderful collaborator in our Department of Biochemistry, Professor Ann Brown. She does a lot of biochemical modeling. And so she looks at enzymes and how molecules dock to enzymes. Now, an enzyme is a lot smaller than the ribosome. And so she has a lot of challenges that she's overcoming right now in terms of understanding how these molecules bind to them. But the process of understanding how these dock is roughly the same, where the antibiotic can be split away from the ribosome co-crystal structure. It can be redocked in order to get a training set. And then we can try to understand sort of how it influences the other antibiotics that bind nearby, make it specific for one certain kind of a ribosome, and then try to redock it to understand if we're succeeding with our training set. One other item that we would like to do as a future work is see if we can optimize a linker. If we could get it to have greater affinity, or if we can't do that, if we can increase some of the other important pharmacological properties without affecting its binding to the ribosome. So I do want to walk you through a discrete example of this. These aren't molecules that we're necessarily working on, but it's a great illustration in terms of trying to understand how this process is going to work. So in the top left portion here, I have sparsamycin shown down here, and the thioester in yellow and blue is in red, and the sparsamycin molecule drawn in ChemDraw. On the right up here is spiromycin, and spiromycin has a sugar that points toward that sparsamycin molecule. And these hydroxyl groups on that sugar are shown here in blue and in green. Those aren't critical for binding. If we're able to put linker portions there, we can move toward making a bidentate antibiotic. So the sparsamycin is fairly straightforward. It's a fairly simple small molecule. We can achieve that using traditional synthesis. A challenge, but not necessarily one that's going to be very difficult to overcome. On the other hand, spiromycin is quite complex. If we buy it and attempt to do a semi-synthesis, we have a lot of protection that we need to do. If we want to do it via total synthesis, that's going to be a lot of steps, especially if we want to get more derivatives in order to put that material together. Doable, yes, but it's going to take a lot of effort in order to get toward that molecule. And this is really where the biocatalysis that I learned in David's group, I think, has a potential to be very, very powerful toward making molecules at this time. We can buy spiromycin, and there's known hydrolysis procedures to remove the mycorrhizas from the natural product. So now we're to something that's in the natural product biosynthetic pathway. Spiromycin is made from neospiromycin. So if we have neospiromycin and we can look at that biosynthetic pathway, we can start to understand some of the enzymes that are responsible for completing the natural product. And perhaps we can use one of those as a biocatalyst in order to get to the product that we want. And so if we were to take mycorrhizas and derivatize it such that we have a proto-linker that we want and the enzyme's capable of accepting it, we can get to our functionalized spiromycin in a much more streamlined approach by using a chemoenzymatic method. Once we have these two materials in hand, we can now dock them together using a variety of linkers. I've just shown some generic examples here in terms of an alkyl, a polyethylene glycol, and of course, click chemistry, which everybody loves to use. And then we'll have a library of these bidentate materials that we can subsequently test against our natural products, or rather against our bacteria. And then we can use the results from that as feedback to assist our computational collaborators in also better refining their models. So again, we're really interested in natural products. We want to figure out how we can make the next generation of antibiotics via semi-synthesis, how we can find new chemical methods that we can use to develop these antibiotic derivatives, and also how we can employ chemoenzymatic synthesis to further derivatize some of these, both toward improving a next generation antibiotic and to synthesizing bidentate antibiotics that we can test them. And of course, no talk is complete without acknowledging the people who are doing the work. As you can see from this collage of group members, you can sort of tell when people join the group. Those who started about 2 years ago have a mask on for these pictures, and the older ones don't. We are fortunate in that we just had our mask mandate rescinded here. Because our vaccination rate is high enough. And so some of our more recent folks have their photo up here without their mask on. Additionally, I would like to thank all of the people who have contributed to my career. So Virginia Tech, first and foremost, for hiring me on as an assistant professor. And then my graduate advisor, Marissa Kozlowski, who got me very involved in total synthesis and loving how to create new molecules, and the work that I did there that really inspired me to go and get a postdoc with Professor David Sherman, who's a member of the CCHF, and learning how to use selective functionalization in order to diversify natural products to give us a bunch of different compounds that we can then go on and test. And so with that, I'm going to put my group members' names up here and show you a picture of the beautiful scenery here around Virginia Tech. And I'd be happy to field any questions orally or certainly in the chat as well. So thank you, Hugh.
Okay, well, thank you very much. Well, this is the time to show your appreciation for the talk, to show that you can handle Menti. That's great to see. Yeah, so very nice talk indeed.